Hello students, a very warm welcome to my YouTube channel. Today we will be talking about northern blotting. Blotting techniques, as you all know, are very important tools in molecular biology. And I will try to discuss each of these blotting techniques in separate lectures so that you don't get confused or it doesn't become too lengthy. Okay, so I'll be dealing with each of them one by one. So let's get started. Today's topic is northern blotting. Blotting is used in molecular biology for identification of proteins and nucleic acids and is widely used for diagnostic purposes. Why do I say here proteins and nucleic acids? Because Western blotting deals with protein. Then for nucleic acid, you have southern blotting. For DNA, northern blotting, that is today's topic. Northern blotting we have for RNA. That is why proteins and nucleic acid. Okay. Blotting techniques involve the separation and transfer of DNA, RNA, or proteins. And how is the separation done? It is done via electrophoretic technique or electrophoresis or gel electrophoresis. Okay. And thereafter, after separation, what is done? It is transferred onto a carrier. What is this carrier? A blotting membrane here, right? The target DNA is then attached to a molecule in order to aid detection. Okay, this detection is done with the help of probes. Okay. Detection is done with probes. All right. Okay. Now we'll just briefly talk about the principle of northern blotting. Okay. Northern blotting is used to study gene expressions okay, by detection of RNA in the given sample. All right. Given sample. Clear? See, the sample RNA is isolated from an organism of interest and then electrophoresis and then electrophoresis is carried out. That is agarose gel electrophoresis is carried out that separates the fragment on the basis of their size. Clear? Okay, so just let's write it. So what did I say? After isolation of RNA, Gel electrophoresis is performed to gel electrophoresis is performed to separate the fragments on the basis of their size. And finally, what is done? The separated RNA fragments are transferred to a support membrane, or as I told, transferred onto a carrier. Support membrane, like nitrocellulose membrane here. This can be performed by, simple capillary method in the presence of a specific buffer. Okay, so this is the basic principle. This is the basic principle of northern blotting. Done? Okay. After knowing the principle, we must know who has actually developed this. So northern blotting is a technique for detection of specific RNA sequence. It was developed by James Alwyn and George Stark. Please use capital A. Okay, James Alwyn, just a minute. Please note this. This is capital A and this is capital S. James Alwyn and George Stark, right? RNA molecules have defined length and are, much shorter than genomic DNA. It is not necessary to cleave RNA before electrophoresis. When you are working with DNA, it is very important to cleave them. Get them into handy small fragments, but RNA molecules are itself small. They have a defined length and they're much shorter. So you need not do the cleaving procedure for RNA before electrophoresis. Okay, RNA is more susceptible to degradation than DNA. So when you are working with RNA, you will have to be very, very careful. It is more susceptible to degradation. And once your sample RNA starts degrading, your experiment will be a big failure. RNA sample are separated based on size by gel electrophoresis. This is what we have just discussed. Okay. Now see, these are the brief steps discussed very briefly here. Total RNA or mRNA is isolated from tissue sample. RNA is separated according to size by electrophoresis through a standard formaldehyde gel electrophoresis or agarose gel. RNA is then denatured in situ. Okay. Denatured RNA is transferred from the gel to a solid support, which is usually a nylon or nitrocellulose membrane via capillary transfer method. Okay. RNA attached to the membrane is hybridized to a labeled DNA, RNA or oligonucleotide probe. This is why it is so very important. It is important for detection purpose. Bands complementary to the probe are located by an appropriate detection system, for example, by autoradiography. So these 7 steps define how simple the process is. It's simple, yet very, very important. Okay? See, this is how the RNA is denatured. As we have just said here, RNA is denatured in situ, and then the denatured RNA is transferred from the gel to solid support. See, the denatured RNA is transferred from the gel to the solid support or nitrocellulose membrane, right? See, look at this. This gives you a better picture of the procedure. Okay. See, you have your RNA of interest here. You run electrophoresis. RNA separated by size, because this is the purpose served by electrophoresis. It will separate the RNA fragments according to size. Then it is transferred onto a solid support. It is transferred onto a solid support, which is the nitrocellulose membrane here. This is the solid support. Solid support. Okay. Then membrane hybridized with radio-labeled probe. Probes are here. Then analysis using X-ray. Okay. So when you are writing a critical note on northern blotting, you will have to write about its, like in the introductory part, you will have to write its definition, the principle, that brief little procedure, and one diagram which supports what you write. Now coming to application, analysis of regulated gene. First and foremost and most important application is analysis of regulated genes. It indicates which tissues express the gene, and it also investigates factors controlling the gene expression. Okay. Next is checking if the cloned cDNA is full length. Comparing the size of mRNA with the size of cloned cDNA. Clear? See, these uses are very, very important. These applications or uses are very important. And from exam point of view, let me tell you, when you are writing the critical note on these topics, you will have to mention about it. You will have to talk about its applications as well. If it comes separately for 2 marks, then at least for 4 applications you need to write. Otherwise, you will have to include this point, this part in your answers only. I mean, in the short note itself, you will have to write it. Okay. So it is really very important for you to know the fact in which fields it is used. Okay. For your convenience, I can write it out also. Often students say that, ma'am, if you write it out for us, it becomes easier. I'll do it for you. Wait, just one. Applications. Okay. Analyze overexpression of oncogenes. Oncogenes, you know, cancer causing genes. Oncogenes and downregulation of downregulation of tumor suppressor genes in cancerous cells. Okay. Next, you can say detection of specific mRNA in sample, used for screening the sole purpose behind writing these out again is to make you write. While you are going through this video, please pause the video and take it down as notes. Just don't gaze through this. It won't help. Practice writing. The more you write, the better it becomes. Please write it. Screening recombinant. Recombinants which are successfully transformed with transfer. Then it also helps in studying mRNA transfer. It can determine whether a gene is transcribed or not, whether a gene is transcribed or not, and also detects transcript size and also detects transcript size. It also helps in studying RNA degradation. Studying RNA degradation, splicing and half-life. Splicing and half-life. Okay. So I hope it is clear to you. This is how we come to an end of our lecture. I'm again telling you, please go through it with full concentration. In case if you have any confusion or doubt, please get back to me in the comment section and stay tuned for more updates. I will be making lessons and videos on other molecular biology techniques as well. So please stay tuned. Till then, logging off. Goodbye.